The following events take place in each archegonium that is fertilized. The diploid zygote nucleus undergoes 2 mitotic divisions, and the 4 nuclei migrate to the base of the archegonium. Successive divisions result in a 16-celled embryo. The tier of 4 cells at the base of the archegonium are embryo initials. The 4 cells of the adjacent tier are suspensor initials. The suspensor initials elongate, first pushing the embryo initials up against the wall of the archegonium, and then out into the female gametophyte, where breakdown of the adjacent tissue creates a corrosion cavity around them. The 4 initial cells separate, and each begins to form an embryo and additional suspensor cells, which push them further into the female gametophyte. Intense competition between the 4 young embryos for nutrients and space results in only 1 embryo surviving. Embryo systems in adjacent archegonia proceed through the same stages of growth and competition. Soon, however, 1 young embryo in the female gametophyte dominates and proceeds to develop further while the remainder abort. This surviving embryo enlarges and develops a shoot apical meristem, surrounded by a circle of seed leaves called cotyledons. Below the cotyledons, the embryo forms an elongated cylinder of cells that represents the stem-to-root axis. The tip of the root contains a root meristem, which in turn is covered by a protective root cap. During embryo development, nutrients accumulate in the female gametophyte, where they will serve as a source of nourishment at the time of germination. The final stages of seed maturation are accompanied by the removal of water from the living tissues, which then enter into a state of dormancy. The integument also undergoes changes, transforming into a tough, protective seed coat. Both the seed and surface tissue of the cone scale detach as a single unit. The mature winged seed is now ready to be released.